Hello, my name is Mike Ward. I'm the head of Informa Pharma Intelligence's Insights publications. We're the publishers of Script, Pink Sheet, In Vivo, and Generics Bulletin. And I'm here at the Biotech Showcase 2019, where pharmaceutical, biotech companies get together with investors to review what's happened in 2018, but also to then look at opportunities going forward in 2019. The 2018 was in fact a very, very interesting time because we saw a record number of new molecular entities getting approved. And, but what we have spotted is that actually the attrition rate for drug discovery and development is still pretty tough. One way of actually mitigating that is actually by going to known drugs, repurposing known molecules that are known to be safe, etc. So I'm joined by Dan Gooding, who's the CEO of a Cambridge, UK-based company, Nuformix, and essentially that's one of the things that you do. You take known entities and repurpose them.
Exactly right. No, you've said it absolutely spot on. I think it's been an incredible year, like you say. We develop known drugs. We have a unique way about how we go about that, and we have applications for the redevelopment of those known drugs based both in generics, but also in your classic kind of reprofiling applications in disease switches.
Right. So how do you identify what you're going to go after?
Sure. So we look for two things. One, unmet needs. First of all, and typically that can be in relation to access to a drug, in terms of patients not being able to get access to a drug, and therefore there's an unmet need there, or in terms of a lack of treatment in a certain space, and where there's been a progression in disease understanding in terms of disease targets or mechanisms. And quite often now we can find that we can retrospectively fit molecules back into new uses that happen to have a very good activity towards the new target. That's, in certain situations, that's a really nice situation, because if we can also look at drugs that have got activity towards that target, they've got a really nice safety profile, that's a very nice situation to have. What enables us to do things slightly differently is we also look for drugs that have got some real problems in terms of how they're delivered. Perhaps they've got very low bioavailability or other kind of problems in relation to how that drug might be reused in a new way. So we look for those kind of opportunities. So our technology, we use it to improve the properties broadly of known drugs. We can also file some very nice and robust IP around the way that we re-engineer those molecules, and then we look to combine those aspects: differentiated drug, very attractive pharmacology, and look to develop the drug onwards from there.
So what is the the core technology that is allowing you to do that reformulation?
Yeah, of course. We're using a technology called cocrystal technology.
Right.
So
what's that?
So with cocrystal technology, we're creating basically a two-component drug substance. Typically, we're crystallizing a drug small molecule with a second molecule, which we call a coformer, which is typically inert and has a safety data package with a regulator body. So, something on the GRAS list or a known excipient or something like that. And in the right circumstances, these coformer molecules can interact with the drug molecule to form a highly stable crystalline two-component substance. And so, because we haven't chemically changed the drug, we just basically crystallize it with a second molecule, the regulatory bodies treat our drug substances as if they're the original drug itself. However, the actual drug substance borrows properties from the coformer molecule, so typically we see an uplift in the kind of properties that are favorable for drug development. So we can create these new substances, get patent protection. However, our regulatory path is really good, and we have some nice differentiated positions for developing them going forwards.
And the new molecule that you have created with your technology, is it going for the same indication that the original drug went for, or are you identifying different indications?
No, we're actually doing both. We've got a lead program, which is kind of a faster demonstration of our technology, if you like, where we're actually making a generic version of a currently marketed treatment in the CIMV space. So it's an oral treatment with a very high sort of technical barrier in terms of development. The drug is actually just recently off patent, but it's been very difficult for generic companies to get into the space because of the high levels of difficulty, both in developing an effective formulation, but also the IP space is very difficult to get around due to the way that the drug's been formulated. And we've got a complete different approach to that. So our first program is actually making a generic version of currently a complex generic. Our second lead program, and more likely the rest of our future programs as well going forward, is more, like you said, the classic kind of disease switch stuff where we're taking a little known allergy treatment and reprofiling it into fibrosis.
And so how were you able to sort of recognize that this molecule actually had potential in a different indication?
Sure, we look for the signs of that, scouring the literature, looking at what's known about what's increasingly known about a drug. Often you find now, such as the pace of research and the breadth of research globally, there's a vast amount of information out there that can tell you, give you signs about what drugs might do. And so we'll look to validate, once we see a concept there around what an old drug might do in a new indication, we'll seek to validate that in the best possible way. And we've just actually done a really innovative study in partnership with Newcastle University in the UK in the area of lung fibrosis, IPF, idiopathic lung fibrosis. There what we've been able to do is we've been able to take tissue actually from patients. I don't know if you know much about lung fibrosis, but it's a high rate of mortality with patients that are diagnosed with it, and one of the best, most effective treatments is actually a lung transplant. So when those patients are lucky enough to receive that kind of opportunity, as their end-stage disease lungs are removed, in a long story short, through work that's been done at Newcastle University, we're able to use that tissue as a therapeutics testing ground. Right. So if you think that disease models in the space are really very poor, so if existing disease models are here and patients are over here, we've moved this model super close to the patient because we're testing our drugs on the tissue that patients are going to be presenting with in clinic. So it's the first kind of large study of its kind that's been reported in multiple tissue donors, and our drug has done really well.
And the business model, I mean, are you looking to sort of commercialize your own drugs, or is this a sort of a platform technology that you would expect other partners to maybe engage with?
I think what we'll be looking at doing is we'll be applying cocrystal technology to a broader range of molecules, and be looking to develop a slightly broader clinical development pipeline. We'll be looking to develop the assets, each individual asset, to a patient proof of concept level prior to out licensing. So, for example, with our lead program, we've already licensed our first lead program, the CIMV program, NXP1. We've already licensed China marketing rights there for that asset. And so as we validate that proof of concept, we're actually performing a pilot bioequivalent study effectively, which will read out in May this year. That will probably be as far as we go with that program. We'll look to out-license from that point, and that's basically our business model, an out-licensing business model after proof of concept.
Right. Okay, well, Dan, thanks very much for stopping by.
Yeah, cheers.